our chemistry seminar from wherever in the world you are. I say warm welcome. It's an exciting morning. You can feel the excitement in the room as these women in science are about to take over the floor. It's an exciting morning for women in science, women in chemistry indeed, as yesterday the Nobel Prize in Chemistry was announced, and for the first time in the history and in the history of the Nobel Prize, a team of women, 2 women only, have won the Nobel Prize in Chemistry. Emmanuelle Charpentier, and she's out of Berlin. She's French and worked out of the Max Planck Institute. And Jennifer Doudna, who is American and worked out of University of California, Berkeley. I mean, we have, certainly as a graduate student, you know, I was reading about these molecular scissors and the technique CRISPR-Cas, which they developed for gene editing. And it offers so much promise for people, certainly in the Caribbean, people who are affected by genetic diseases like sickle cell. So this morning, these women, the 6th, I think, and 7th woman who have won the Nobel Prize in Chemistry, we are celebrating with them. And it's fitting this morning that we have outstanding women, young women like Dr. Catherine Murray, who will be talking this morning in our seminar. So we're celebrating women in science. And the other thing we are celebrating, and you could have felt the energy when you walked in and you saw the reunion happening between Kenisha, Catherine, Rashida. We also have Henry, and I think Mark is here. All of Dr. Pinnock's, Willard Pinnock's past students, not in recent times, the recent ones, are here with us. And this morning, fittingly so, we have decided to do these seminars in honor of Dr. Pinnock. Without further ado, I'm going to hand over to Dr. Kenisha Wilson. And Kenisha is herself moving some mountains over there in the Middle East, in the United Arab Emirates at Zayed University. Kenisha is doing wonderful things, and you will hear from her in a couple of weeks' time when she also gets her chance to celebrate with her work Dr. Pinnock and his contribution to her life. But Kenisha this morning, Dr. Wilson this morning will introduce Dr. Murray. Dr. Wilson, over to you.
Thank you, Dr. Wilmot. Finally, I guess it's the excitement is tangible. It really is. And before I introduce Dr. Murray, I'd just like to thank you for this opportunity. It's been a while since we have gotten together in any sort of forum. So this is really exciting and refreshing. And I'd just like to thank you and the entire chemistry department for facilitating this. I mean, we have a lot to be thankful for. That's where the foundation was laid that have enabled us to, you know, spread our wings and be confident in going outside of UWI. So today it's about Catherine presenting. Catherine did her bachelor's at UWI in Cape, and she did a major in chemistry. A little bit later, she decided she wanted to do an MPhil, and so she joined us on the Mona campus. So that's where the group was formed. It was Catherine, Mark, Rashida, Henry, and myself. And we had some of the best years of postgrad during those times up in our little office in the environmental lab. Yeah, the best of times, the worst of times. But I think at the end of it all, we've certainly bonded and we have remained in contact over the years. After Catherine finished her MPhil, she decided that she wanted to pursue a PhD. And so she went further afield to the Nottingham Trent University in the UK, and she completed a PhD in analytical chemistry. After that, she switched gears a little bit, and she spent over 7 years in research and teaching of GI imaging, gastrointestinal imaging, at the Sir Peter Mansfield Imaging Centre in Nottingham. There she used MRI to visualize the GI tract in health and diseases. Dr. Murray's research interests lie in gut health and the gut microbiota. which recently led to a new position at the Quadrum Institute Bioscience in Norwich, same place in the UK. She's currently investigating the effect of dietary plant phytochemicals on the diversity of our gut microbiome. So today she'll be sharing some of her outstanding work and publication with us as she shows the key time. I think, was I muted for a little bit there? Did you get the last part of what I've said?
No, we didn't.
Today, Catherine will be presenting some of her outstanding work, a few of her papers that she published in high-ranking journals on the key interface of chemistry and medicine, how these overlap. And she'll be showing us simple processes of eating and digestion, and to cite some examples of how these things can go wrong. So this is really a presentation that I'm looking forward to, because it shows you the applicability of chemistry as we go over into different types of disciplines. So could we just welcome Dr. Catherine Murray to the floor at this time, please?
Thank you. Thank you very much, Dr. Wilson. And I want to say thank you also to Dr. Singh Wilmot for facilitating this. I am honored to be back at UWI Bona. I just look back at my time there with great fondness, and I remember everyone was just so welcoming to me. When I first started there, the others in the group just took me under their wing, and I just felt like one of the rest of them, even though they teased me mercilessly about the size of my island country. I'm from Barbados, but yeah. So Dr. Pinnock himself also joined in the teasing occasionally. But when I first got there, I remember Rashida described Dr. Pinnock as a big teddy bear. And when I met him, I agree with what Rashida said. He was just so warm and kind and fatherly. And like a good father, he ensured that we gave of our best. And that translated into our best in terms of writing, in terms of research. I know Dr. Pinnock was a stickler for good research, and he would question the validity of some things. If you wrote something that didn't make sense, he would let you know it didn't make sense. And I want to sit here all these years later and just thank Dr. Pinnock for that background, because I use the Pinnock method myself on my students. And so I require a certain degree of excellence from them in terms of writing. And so I'm just going to share my screen. Hopefully it's going to work for us. Can you see my screen?
Yes,
okay.
We can.
And so I think the 5 of us in the group, Kenisha, Mark, Rashida, Henry, and myself, we can all say that our lives have changed somewhat. We have been improved for having worked with Dr. Pinnock. And in the words of Sir Isaac Newton, if I have seen further, it is by standing upon the shoulder of giants. And I think that we would all agree that we have stood on the shoulder, we're standing on the shoulder of giants such as Dr. Pinnock. And I think I'll just mute, take my camera off to save my bandwidth. Okay. But I'll keep talking. So as Kenisha said, I spent some years working in, years working in the Sir Peter Mansfield Imaging Center, where I developed a love for digestive diseases and disorders, and we use MRI to image these disorders. And I think it's important for me to say that if you are squeamish, if you have decided to have an early lunch, perhaps you would not do that, because this is all about digestion and digestive disorders. And I've chosen this background because this is quite similar to the sort of weather we get in the UK on a regular basis. There's just lots of clouds. The sky is actually bluer there in this picture than it is normally in the UK. But moving swiftly along, I've got about 45 minutes to present to you my work. So just an outline of what I'll talk about. I'll give you a brief overview of digestion and a brief overview of magnetic resonance imaging, which is the modality which I use to collect my data. And then I'll present a few of my papers, just a few of the ones that I've published where we do some gastrointestinal imaging studies, and then I will conclude. So moving swiftly along, an overview of digestion is a really simple process. As we know, we take the food in our teeth, they bite and begin the process of mechanical digestion. Our salivary glands release amylase and linguolipase to help with the initial breakdown, the chemical breakdown of starch and fat, respectively. And it's quite interesting that fat is actually doesn't start breaking down in the mouth, because the pH of the mouth is actually not optimal for activity of linguolipase. And let me just get my pointer out. Pointer. And then the food, it goes down through the esophagus into the stomach, where we have stomach acids being released to further help with the digestive process. And then it's sent into the small intestine, where we have further enzymes added from the pancreas to help with the digestive process. We then have nutrients being absorbed there in the small bowel, and after nutrients are absorbed, we basically have waste, which is eventually expelled from the large intestine. And when we look at digestion, at any point along this digestive tract, anything can go wrong. So starting in the mouth, we could have a situation whereby there is no saliva produced, and therefore we don't have amylase or linguolipase being produced in the mouth. Or in the esophagus, we could have the lower esophageal sphincter malfunctioning such that we have acid from the stomach going back into the esophagus, which we all know acid reflux or heartburn, more commonly called. In the pancreas, we could have blocked ducts. In the small and large intestines, we could have motility issues. We could have overgrowth of bacteria. There's any amount of things that could go wrong in the gastrointestinal tract. But what my research focused on was the functional disorders of the digestive tract. And what I mean by functional is those disorders for which there is no visible problem. A person would go to the doctor with this complaint, and the doctor would run tests. They would take images using endoscopy or colonoscopy or whatever, and they would see absolutely nothing wrong with them. And this is where our research capitalized on the ability of MRI to get inside the body non-invasively and show us what is actually going on and help us create a picture of what is happening there. And so just to give you a brief overview of MRI, Dr. Wilmot was talking earlier about Nobel Prizes in chemistry. Well, Sir Peter Mansfield, who I had the privilege of working with, I didn't work with him directly, but I worked in his building. He was awarded the Nobel Prize in 2003, a prize which he shared jointly with Paul Lauterbur from the United States. And this prize was awarded for his work with magnetic resonance imaging. He did not specifically invent MRI, but it was his work along with, independently, but simultaneously with Paul Lauterbur, which allowed us to create an MR image from the signals which are produced. So I'm pretty sure that each of us, all of us in this audience, knows about NMR, and we can think of MRI as a spatial map of an NMR active nucleus. So in the simplest of terms, we have protons in various environments in our bodies, and when we apply, we put them in a magnetic field and they all line up in the magnetic field. Then we apply a pulse, which causes them to resonate, and as they return to their initial positions, they emit a signal which is captured by a receiver coil. And the rate at which they emit this signal differs based on whether it's water, fat, bone, blood, cerebral spinal fluid. And in that way, we're able to build up an image of a person's body. And as I said, Sir Peter Mansfield was instrumental in creating this transform between a frequency signal and the image, because we have what we get from the NMR-like sequences is basically called K-space, and it's a series of dots on a screen. but a fancy mathematical calculation, which Henry could probably help us with, but I think it's beyond the scope of my own personal understanding. There's a Fourier transform calculation, which gives us the image. And so this is how MRI works, a basic overview. And just to give you an example of what it might look like in the bowel, so this is the coronal image of a person who has just had a meal. So this is their liver, this is their gallbladder, this is the stomach, this is the heart and lung region, and then further down we can see their small intestine, and we can see matter being pushed through the small intestine. And an image like this can tell us quite a bit, because we can see particularly how motile the parts of the gut are while they're working to digest food. So if the person had issues with motility, then we wouldn't see all this action of the stomach. So this is just one example of a healthy participant after eating, and we can see the sort of information that we can get from the MR image. And so moving swiftly along to how I use MRI in my studies. So I'll begin with one of the first studies I did when I started working at the Sapita Mansfield Imaging Center. And this was alongside Unilever. So they used to market Slim Fast, but they've subsequently sold Slim Fast. But as we know, obesity is a massive issue, and Slim Fast was designed to help people with weight loss. And I think obesity is probably more of a pandemic, but I didn't want to use the P word because of the present time that we live in. But nevertheless, it's an issue that affects us worldwide. And one of the main reasons for obesity is an overabundance of meals that are energy dense, but have low appetite value. And so things like Slim Fast and Weight Watchers programs have been designed to help people lose weight, but these things are all subject to consumer compliance. And so a person, for example, who's on Slim Fast and they eat the shake, they drink the shake or the meal and they think they're still hungry, will have another meal. And in that way, they would not lose weight, they might actually gain. And so there is a need to prepare healthy meals that deliver maximum satiety, but for minimum calories. And this way you would reduce the feelings of hunger between meals and reduce snacking. So the guys at Unilever noticed that when you introduce air into drinks, things like a foam, if you eat a mousse, you might feel more full than if you have a drink, or if you have something like a can of Coke, you might feel more full than just water initially. And so they wanted to know the mechanism whereby aerated foods would increase satiety and reduce the need to snack. And so they came to us and they wanted us to study their foam drink. So they wanted to create a foam version of Slim Fast. I'm not sure if they've actually done it, but I do know that they sold it shortly after this study. And so the first step was for us to look at in vitro models of foam decomposition and foam collapse. And so this was a foam whipped up by a simple cake mixer, and this is about 850 milliliters of foam, and we left it on the bench over a 2-hour period just to see how the foam collapsed over time. And we can see after 2 hours, it was just about 200 milliliters left, as opposed to the initial just under a liter. And the question then would be, how does it behave in a stomach, a human stomach? And to answer that question, we recruited 18 healthy participants, and they came on 3 occasions. We say the study was partly blinded, and it was only partly blinded because participants had on 1 occasion a drink which was not a foam. So they would have known when they were getting the control drink. But then there were 2 foams which they wouldn't have known whether it was the stable foam or the less stable foam. So these 3 drinks were equicaloric, so they all contained the same number of calories. 1 of them was just the liquid, and 2 of them were whipped in a cake blender, sort of a cake mixer, to create the foaminess. And so they were whipped to about 450 ml, while the liquid control was just about 200 ml. And so after an initial baseline MRI scan, we then had the drinks fed to them, and then we scanned them over a 4-hour period using MRI. Simultaneously, we collected appetite ratings. So they were asked, it was an electronic visual analog scale. So basically they were asked, how hungry are you? And questions like, how willing are you to have another meal? And they had to rate their symptoms, their feelings from 0 to 100, 0 being I'm stuffed, 100 being I'm starving. And this is an example of our scanner. It's a little magnet. This is the 1.5 Tesla magnet. And unfortunately, it became an unreliable way of collecting data, so it's no longer in use. But just to show you the participant position, so participants were positioned supine, and you might wonder how physiological that would be lying down to take an image of digestive processes. But studies have shown that there's very little difference between collection of images while they're seated or lying down. And between images, they were seated in normal upright position. And they have a receiver coil over the abdominal region to collect signals from the protons in that region. So just to show you the results, initial results. So these are pictures of a participant on the left of the screen. This is 10 minutes after drinking the stable form. And on the right of my screen, it's 90 minutes after drinking. And we can see that the foam layer decomposes over time and the stomach volume decreases over time, which is exactly what happened in the in vitro study. But when we compare the stable form to the less stable form, you can see that there's about 20 minutes between these two images, and the stable form doesn't break down quite as quickly as the less stable form. You can see after 30 minutes that there's a lot more liquid in the stomach after consumption of the stable form. And indeed, this decrease in foam content was significant compared to between the two foams. So looking at a total time course of the gastric volume, this is the time course for the liquid control, and the gray is the less stable form, and then we have the more stable form, the black circle. And we can see that, as expected, the liquid control, with it not being wet, produced a lower gastric volume, but incidentally, all three of them left the stomach by 2.5 hours after they were consumed. So compare the total gastric volumes to hunger scores, so we can see a relationship immediately. The young men felt a lot more hungry when they had the liquid control drink as compared to the stable form and the less stable form. But what was interesting for us, and something that we couldn't quite figure out, is that despite the stomach emptying after 150 minutes, we still see a departure from between the stable form and the less stable form in terms of hunger. And so while we could argue that the reason for satiety in the earlier part of the study was a result of the total gastric volume, we can't really use gastric volume as a reason there. So there is some mechanism operating within the small bowel perhaps, or within the stomach after, I don't know, maybe a stretch mechanism, which is allowing people to feel satisfied after the fat, even after the stomach has emptied. And unfortunately, as I said, SlimFast was sold, so we couldn't do any further work on that. But MRI was able to give us some idea of the processes of digestion and help us to see how we could potentially create foods which would allow people to eat less, but still be able to lose weight by feeling full longer. And the beauty of MRI, while it's a very simple study, I will not say that it's a complicated, it's a very, very simple study that we carried out. But the beauty of it is that MRI can non-invasively measure physiological processes in the non-disturbed bowel. So normally, in order to measure processes, people would use maybe a sort of endoscope or colonoscopy method, whereby you could put a tube in from the top or the bottom to see what is going on inside. However, with MRI, the tube doesn't have to be there disturbing the bowel. We can see what is happening in real time by just a simple click of a button. So it's just the beauty of a physical chemical method applied to medicine, which gives us all these results. So now moving on to irritable bowel syndrome. As I said, if you're squeamish, apologies in advance. And this is another functional disorder. So as I said, they're disorders whereby you can't see a reason why the participant is suffering. And this accounts for a really high percentage of referrals from the primary care physicians to gastrointestinal outpatient clinics. And this disorder is characterized by disordered bowel habit. And by disordered bowel habit, I mean it can swing from constipation to diarrhea to a mixture, depending on what type of irritable bowel you have. And there's abdominal discomfort, such as bloating and pain sometimes. It's also associated with stress and anxiety. In the Western world, women tend to suffer with it more than men. But I'm told that in places like Asia, you tend to get more men suffering with it than women, but who knows why. And several patients tend to associate their symptoms with some foods, and as a result, many will restrict their intake of some foods to help them ease their symptoms. And one of the really popular diets you might have heard of is the FODMAP diet, and this is a very restrictive diet. So what are FODMAPs? So that is actually an acronym which means fermentable oligosaccharides, disaccharides, monosaccharides, and polyhydrate alcohols, or also known as polyols. And it's basically a collective term applied to a group of short-chain dietary carbohydrates that are poorly absorbed, so they don't get absorbed in the small bowel as they should do. They're highly fermentable, so they go through to the colon where bacteria which are present there can break them down. And they're osmotically active, which means that they have the ability, as we all know about osmotically active particles, they have the ability to pull water into the lumen of the intestine, thereby creating perhaps a diarrhea-like condition in some people. And so some examples of these FODMAPs, fructose and fructan, they're the common culprits in our diets these days. Fructose, as we know, is a monosaccharide, and fructan is a polymer of fructose in the oligosaccharide form. Then we have disaccharides, such as lactose. We have other oligosaccharides, such as galactans. And finally, we have our polyhydrate alcohols, which are things like sorbitol, mannitol. But as I said, fructose and fructans, those are the main culprits in today's world. So just to show us what types of foods contain FODMAPs, we all know that fructose is obtained from fruit. But perhaps we didn't realize that there's a very high concentration of fructose present in dried fruit. And then it's commonly masked under the name high fructose corn syrup in a lot of canned and preserved products. So we can easily get lots of fructose into our diet. And then the others, such as oligosaccharides, they're the more complex carbohydrates. So you'll find them in things like your grains and your pulses, legumes, and your glucosinolates, such as broccoli and Brussels sprouts. And then our disaccharides, such as lactose, we would find obviously in milk products. And then the polyhydrate alcohols form the basis of sugar-free sweeteners. So you'd find them in things like sugar-free gum. So what is the theory behind this FODMAP diet? And why is it that some people have symptoms as opposed to others? Well, we can't answer yet the question why some people have symptoms other than, unlike others. but we can give you the theory at the moment. So fructose in the diet, and we're talking about a large amount of fructose, not a small amount. Fructose that cannot be fully absorbed. And some people can absorb fructose, but some people have the issue if they eat too much fructose. And this is poorly absorbed in the small bowel. And because it's osmotically active, there is water delivery to the lumen of the small bowel. And the excess that is not absorbed is delivered into the large bowel or the colon, where the bacteria which are naturally present there fermentate and release gases, which are normally hydrogen and methane gas. And these gases produced lead to the abdominal discomfort and altered bowel habit, which sufferers of irritable bowel syndrome tend to suffer from. So fructans, the other main culprit of in today's diet, they are not osmotically active. So you don't get that watery delivery. Well, you wouldn't expect the watery delivery through to the small bowel, but they're also not absorbed in the small bowel. So they're all delivered to the colon. And then you get the same activity whereby you have bacteria breaking them down and there's a release of gases. But then the theory goes on to say that when glucose is in a one-to-one relationship with fructose, or when the concentration of glucose in the meal exceeds the concentration of fructose, then we have a situation whereby fructose absorption is improved. And one of the reasons for that, again, it's a theory, so some people would not agree with it, but the theory is that the transporters for glucose are also able to take fructose, but obviously they wouldn't take them in the absence of glucose. So when we put glucose with fructose, we have a situation whereby more fructose is absorbed, and therefore you don't get any of these symptoms or watery delivery to the small bowel. And this is the reason why after eating foods where glucose and fructose are in a one-to-one ratio, or glucose is in excess, you wouldn't expect any symptoms to be observed. So these sorts of foods include things like citrus fruit, bananas, and it's not uncommon for glucose to exceed fructose in overripe fruit. So things like a very ripe apple, you probably would get away with in terms of fructose load. So we wanted to do a FODMAPS study. Again, a very simple study. It was probably one of the simplest studies I've ever done, and it's one of my most highly cited papers, and my first project student did most of the work. So we wanted to test the hypotheses that fructose would increase the amount of water present in the small bowel more than fructans. And we wanted to see what would happen when we added glucose to fructose, but we hypothesized that addition of glucose would reduce the amount of water collecting. And we also hypothesized that fructan would have very little effect in the small bowel since it's not osmotically active, but it will exert its effect through colonic fermentation and the release of things like hydrogen and methane gases. And just to give you a very quick background on how we look at water content in the small bowel. So as we know, MRI, I don't know if I mentioned it, but I probably should mention that we do not, at the MR Center, we did not do any imaging with contrast agents. So I know contrast agents are commonly used to highlight differences in various structures, but we simply got different images based on the fancy imaging sequences. So for example, this particular image, this is a coronal image of a person. We're looking at their abdominal region. So that's their gallbladder, the stomach, and this is the small bowel region. And what we did was we created a heavily T2 weighted sequence, which basically is biased towards picking up free water. So any water that's bound, for example, in tissues, it wouldn't pick up. So everything that doesn't have free water looks completely black. And then we're able to detect what is free water. And we would expect that the water produced by the presence of fructose would be free water. And we do this by lengthening the repetition time. And the repetition time is basically the time between if you're taking multiple pulses, it's a time between subsequent successive pulses. And then the echo time is the time between the application of the excitation pulse and the peak in the measured signal. So we can make these particularly long so that the only thing that shows up is free water. And we've got a brilliant scientist at the MR Center called Caroline, another woman who's a brilliant physicist. And she validated a method whereby she compared a person drinking water to her semi-automated extraction and quantitation method. So a person would draw around the region that they're interested in, and what you end up with is just your region of interest, which, using her fancy calculation, will give you the volume of water in that region. So we use that to determine how much water is present in the small bowel. So how did we go about doing all of this? Well, once again, we recruited healthy participants. We repeated the study in patients, but I won't show you that data here. That data was quite interesting though. So we recruited 14 healthy participants. They came on 4 occasions, and each time they had about half a liter of water, which contained either glucose, fructose, fructan, or a mixture of glucose and fructose in an approximate 16% concentration. They had a baseline scan, and then they drank the drink, and then they were scanned hourly for a period of 5 hours. So once again, we scanned them on the 1.5 Tesla scanner. That's Victoria, the project student I mentioned. And we can see here that she's put in ear defenders on the participant. The scanner is very loud, so we give them ear defenders, and we also give them earplugs to help protect their hearing, basically. And after each scan, we measure the amount of gas being released by bacterial fermentation in the colon. And while I said it could be hydrogen and methane, it was easier at the time for us to measure just hydrogen. So that was what we did after every scan. And we collected that data by a simple handheld hydrogen meter, which gave the amount of hydrogen present in an expelled breath in ppm, parts per million. At the same time, we asked them to complete questionnaires, asking them things like how much pain you're in, how bloated you feel, that sort of thing. And so results, remember we said we wouldn't expect anything to happen with glucose, and as expected, we can just see that the small bowel water scans only showed what's in the stomach, the gallbladder, but very little is showing up in the small bowel, which is a good start. But then we got really excited when we saw what happened with fructose, because look at all of this water. That's just proving what everyone was saying. Fructose is osmotic, but this was the very first time in history that, I'm getting excited again, this was the first time that we could see what fructose actually did in the small bowel when we gave a person a large dose of fructose. So you can imagine that if you have a lot of dried fruit or a lot of high fructose corn syrup, and this is what's happening in your small bowel. When we added glucose to fructose, remember we expected glucose to mute the activity of fructose because fructose would then be better absorbed. And so we're not seeing much water collecting in the small bowel. And then with fructan, in this particular volunteer, there was more water than we expected to see, but still not quite as much as the simple sugar fructose. So looking at the time course of all 16 participants over the course of the study, we can see that fructose significantly increased the amount of water in the small bowel. And when we added glucose to fructose, this amount of water decreased. Unfortunately, that decrease when we added glucose was not significant, but we suspect that was just a matter of the size of the study. It was too small to actually pick up the differences, but we were quite pleased with that result. Looking at breath hydrogen, so we said that we would expect fermentation to occur as a result of unabsorbed fructose going through to the lumen of the large intestine, and this happened, so we can see the spike in breath hydrogen, and then it reduced over the course of the day for fructose. Whereas with the fructan, which is all delivered into the large bowel and there fermented, then we could see that it rose steadily throughout the day. And at the point where we ended the experiment, it was still quite high. Obviously, if we continued, it would have come back down eventually. So these results were all very positive, but what was quite fascinating, we looked at the bowel of a participant at baseline. So this is their ascending colon, the bottom bit, transverse colon, and we can see very little because they've been fasting. Compare that to the colon 255 minutes after drinking fructan. We can see bits that weren't even there at baseline, simply because the colon has blown up due to the amount of gases produced by the bacteria that are present. And it's interesting to note that participants did not really report many symptoms. Some of them said that there was mild feelings of bloating and gas, but there weren't many reports of symptoms despite all of this. So I said that we repeated this in patients, and patients, they tended to say, I'm feeling pain, I'm feeling bloated. So there's obviously a specific mechanism happening between the brain and the gut of a patient, which causes them to feel things more so than a healthy participant. So looking at the amount of gas produced, and we measured gas by simply just drawing around bits of black, regions of black within the colon. And we could see that fructose produced some rise in colonic gas, but fructan produced quite a large rise compared to other drinks. There was no significant difference between fructan and fructose, which was quite interesting. But when you added glucose to fructose, there was a significant difference in gas production compared to fructan. So that shows that glucose did indeed improve the behavior of fructose. So as I said, only minor symptoms of abdominal discomfort were reported, but those reporting symptoms of gas, we could see a correlation between the volume of gas and their symptom. And going back to the patient study, one patient said, I am happy to see that I'm not actually crazy, because unfortunately a lot of physicians just think that people with irritable bowel syndrome are crazy and they just write them off. And so patients were happy to see that it's not all in their head, it's actually something that is affecting them. And so to summarize, we did see increased small bowel content. With fructose, we've got a rise in breath hydrogen for both fructose and fructans, but as we saw, the breath hydrogen effect was more pronounced with the fructan. The addition of glucose to fructose reduced breath hydrogen significantly, but this reduction was not significant with small bowel, even though there was a tendency to reduce the small bowel water content. And so I'm not sure how I'm doing for time.
We have enough time, Catherine.
Yeah. Okay, all right.
I'm doing well.
Okay, great. So moving on to another problem, which is quite dear to my heart, because I have worked quite a bit with constipated patients, and I've learned to see that they're suffering quite a bit, partly because it's a painful disorder, and also because it's an embarrassing disorder. So it's not something you would speak about at, like over dinner, like you would talk about a heart condition. But it affects 1 in 7 adults and about 1/3 of children at any given time, according to the National Health Service in Scotland, their publications. And for some people, this is a simple issue, so they can do some exercises, they could probably take some probiotics, they can they could have a laxative and it would resolve. But for others, it's a chronic condition known as functional constipation. And what can MRI tell us about this condition? Well, we can learn things about the time it takes for things to transit through the bowel, which will give us an idea of if there's a blockage of some sort. It can tell us about how what we're giving them is helping them. For example, is it making the colon bigger or smaller? Is it making the colon wet or not? And so quickly just going through this, these are some transit pills which we designed in our group. You can see, I'll pause this one on the, if I can. Okay, well, let's just look at the left for now. We can see these large pills just at the bottom of, these are the bottom of the sigmoid colon. They're about to come out of the body basically. And these are gadolinium filled, and we give them to participants to measure how quickly things move through the body. So we would give it to them and see after 24 hours how far along the bowel they've moved. And we can see here that they've moved all the way through the bowel and now they're about to come out. So this is a healthy participant here on the left. On the right, this is a patient with chronic constipation. This is also 24 hours after having swallowed these tablets. And we can see that they're nowhere near the bottom. They've made it somewhat through the intestine, so they've gone through the transverse colon and they're now on their way to the descending colon, it looks like. So this is going to take another few days before they come out. And some participants, even after 48 hours, you still see it there. So this is a good way to use MRI to non-invasively see, is a person, is food transiting through the body as it should do, or is there a blockage? Is it because things are too hard? Is it because, what reason is it? So this is a good way that MRI can look at constipation. Another way is to look at the volume of the colon. So on the left, this is a colon which is in its normal state, so it's quite a thin colon. On the right, this is after it's the person has had ispagula, which is a fiber, like a laxative drink. And we can see here that ispagula is a bulking agent. So we can see here that there's a lot of bulking going on in the colon, and that helps us to see that the laxative is actually working for the patient. Similarly, the amount of water being produced, there are some laxatives that are secretagogues, so they cause the production of water, or they're osmotic laxatives, which again cause water to be pulled into the lumen. And we can see how the laxative is working by doing an MR image of the colon. And this is the ascending colon without the laxative. This is the ascending colon with the laxative. And finally, T1 measurements. So we know that T1 measures the tumbling of water molecules. And we can see that after having a laxative, constipated persons have a longer T1, and that indicates that there's more wetness there in the colon, which is indicative of production of a stool which is not hard like a constipated stool. I'm sorry for the very descriptive methods. So this is another way that MRI can show us about the colon. And finally, before I finish, I'm running out of time here, childhood constipation, very dear to my heart, simply because for children, it's difficult to manage. Children suffer more than adults because when the colon is so backed up, you might get overflow, which causes them to leak and smell, and they struggle with life and constipation. And unfortunately, there are so many unsuitable treatment options simply because children are not just small adults, but we seem to treat children as small adults. So this method is the radiopaque marker method, which consists of them swallowing 24 radiopaque markers over a 3-day period, well, 24 each day for 3 days, and then they get repeated X-ray scans. X-rays are not good for children. They're not good for women of childbearing potential. And not only that, but this is not a good method for seeing what's wrong with the child because we can't tell where the X-ray marker is. So my group came up with an alternative, which is called TransiCap. And this is the gadolinium-filled pill that I showed you in an earlier slide. And this is TransiCap, which is a very small pill which we developed for children with constipation. And you probably don't know what a 20p coin actually looks like. So this is it in the hand of a normal-sized man. And we give these to children. We image them in a large scanner, which does not give them radiation. It has nice mood lighting. And so it's much more comfortable for a child. And we could actually see it in the rectal region of a small participant. So they're working, and there's a big trial ongoing in Nottingham at the moment with this and children. And unlike this, which contains gadolinium, these small ones contain fat and water in a water and oil emulsion, which I designed. And this allows you to image in multiple modalities using the MR. You can do fat suppression, water suppression. So to conclude, What really happens when we eat? Well, a number of complex processes happen. They work in harmony to help us digest what we've eaten. Unfortunately, sometimes it goes wrong, but MRI gives us the ability to visualize these processes and help us work towards improving things when they go wrong. So I hope I've given you a whistle-stop tour, which has shown you a bit more about what I've been up to since leaving UWI Mona. I'll just give you a few of the main names in the team. Caroline, who I said is a brilliant physicist, and most of the work was designed by Professors Marciani and Spiller, and these are all of our collaborators and sponsors, etc. So thank you very much for listening, and in keeping with what today is all about, and what I said at the beginning about me being able to be a better scientist because of working with Dr. Willard Pinnock, I want to say to him, thank you as well. And that's it. Thank you.
Thank you very much, Dr. Murray. And I am sure Dr. Pinnock is in heaven looking down and feeling very proud of you, as we all are at this moment. I am opening the floor, colleagues, to questions and comments. I want to acknowledge the presence of, I see Carolyn, who is one of Dr. Pinnock's daughters. I don't know if she was able to join us at the beginning to hear when Catherine spoke about the Pinnock method that has guided her life, and the shoulders of that giant on whom she stands. I see we have colleagues from St. Augustine. I see Michael Ford somewhere. I see Dr. Gajada, who is in St. Lucia. I had sent the invitation also to Cave Hill so that they too can come and share in who you have become. But I'm sure there are a lot of questions because this thing impacts all of us, this groundbreaking work that you, another outstanding woman in science, is taking on. And so I invite colleagues to just raise your hand if you have a comment or a question, if you just want to say, keep on going, Catherine, or give your own salute to Dr. Pinnock. I do have a question, so I can begin while people get their questions together. We do have time for a robust discussion, and it would be a shame to have her go without having that kind of interaction. So Catherine, it's the gadolinium complex they're using, gadolinium DOTA, gadolinium DTPA. Can you tell us a little?
So it was DOTAREM, which is, I think that's the porphyrin ring that holds the gadolinium. And you're probably aware of the issues with gadolinium and like the kidney buildup, but I've been told that the porphyrin ring doesn't really do that. But we're moving towards phase one. Yeah.
Okay, and I don't see any hands going up as yet, so I can continue. The pre-treatment of the patients. Mm-hmm. How do you pre-treat these patients? I mean, and the selection of the patients that you use in your study.
So I've only presented healthy participants. So in order to get a healthy participant, we just say we want people who have no known bowel conditions. For patients, we have quite a rigorous method. There's something known as the Rome criterion, which basically asks a series of questions about bowel habit and abdominal discomfort after meals. And it is able to select patients who have functional disorders. More often than not, these patients would have spent quite a bit of time going back and forth to the doctor's surgery. So they would have had any amount of biochemical tests. They would have had images done in terms of like colonoscopy and stuff, and they wouldn't have seen any issues with them. So they would have been managing symptoms at the point. So it was actually quite easy to get participants because there are a lot of people suffering, unfortunately.
All right, I see some comments here in the chat. Dr. Murray, I see Dr. Gajada saying it is very exciting work. Dr. Ford said he had to leave, but he thinks it was a great presentation, very informative. And Carolyn, Dr. Pinnock's daughter here, is saying, I'm absolutely fascinated. I'm Carolyn's daughter, and I'm a pediatric surgeon. I do a lot of work with children and with constipation. And in Jamaica, we have just started doing marker studies. I would love to follow up with how these new markers work.
Oh my gosh. Thank you so much. Do give her my details, Dr. Wilmot, and we can definitely contact each other. Oh, wow. This is an honor. Thank you so much, Dr. Jackson.
Wonderful. Dr. Jackson and Dr. Murray have been brought together by you-know-who.
Mm-hmm.
I see someone has, let me see. I think someone has opened their microphone. No, I don't see any hands up. Anybody else who wants to make a comment? I don't see any.
I've noticed in the past that this type of talk doesn't stimulate lots of discussion. It's just one of those things people listen to and they go, okay, that's nice. So,
I think we were all just stunned with the brilliance.
Oh, stop it.
Rashida, did you have a comment or a question?
It's always great to see you, and your work is brilliant. You mentioned in your research that irritable bowel syndrome, a lot of physicians, put it under the table. And that it's not
I'm not hearing you properly.
I'm sorry, you hearing me better?
Yes.
Yeah.
Right. I'm saying that you mentioned that a lot of physicians don't take irritable bowel syndrome as a serious thing.
Mm-hmm.
Now that you have proven that there's something going on, even though the exact mechanism is not there, have you been able, or have your group started to sensitize them that this is actually something that is really affecting people for more research to be done?
I think the quick answer to that is yes. Not just with my work, though. There's actually an irritable bowel syndrome conference every 2 years, so believe it or not. And I think slowly but surely people are realizing that this is not a small issue. So what the physicians who say that people are crazy, it's generally the family medicine doctors, whereas the gastroenterologists tend to be the ones to follow up and say, this is actually a problem which we're seeing in our clinics. It's more than just a psychological or psychiatric disorder. And so, yes, there's a lot more research going into it. And unfortunately, a lot of the data is coming out saying, oh, so now we know more about it, but we still don't know how to heal it, so to speak, or to prevent it from happening. But yeah, a lot of more research is going into it now.
Anybody else would like to jump in? I see Dr. Abdul Rashid, who is joining us from Canada. Okay, Dr. Minot Cates, your hand is up. You may go ahead. This is our head of department, Dr. Donna Minot Cates. Dr. Minot Cates, we lost her. I have Dr. Minot Cates, and then I have Mark with.
Sorry, are you hearing me now?
Yes.
Yes, we are.
Yeah.
Oh, apologies for that. I was just saying, Dr. Murray, that I was fascinated with your entire presentation, and thank you so much for that. I am currently lecturing carbohydrates in food chemistry, and one of the things that we talk about is in terms of the dietary carbohydrates and their impact on health. And I had just quoted a study to my students, actually, on fructose, in which the dietary fructose had been shown to alter the composition, localization, and the metabolism of gut microbiota, with worsening colitis. So I am happy now to be able to quote your work to them and say, here is a product from our department, someone of whom we can be justly proud for this really groundbreaking work.
Wow.
And I know that Dr. Pinnock is here. So thank you. I just wanted to make that comment. It wasn't a question, but congratulations. We are extremely proud of you, and thank you.
Wow, thank you so much, Dr. Minot-Cates. I really appreciate that. Thank you.
Okay, thank you, Dr. Minot-Cates. We have Mark, and then we have HGL. I'm not sure. HGL will have to introduce themselves to us. But Mark, you go ahead, and then HGL can jump in.
All right. Good afternoon, Dr. Murray. Thank you for that presentation. Are you able to hear me clearly?
I can hear you clearly, Mark. Thank you.
Okay. I noticed that the baseline, let's say the participant was fasting. You had said that they were in the fasted state.
Mm-hmm.
And then you gave them the carbohydrate, and it produced such a gaseous response in terms of the H2. I'm wondering if that's the same response if when a person breaks their fast, they eat, let's say, a fatty food. And the reason I'm asking this is, you know, a lot of persons engage in various fasting fads, for example. And when the first meal that they would have is, let's say, a heavy carb meal, is that a dangerous thing? Could that be alluded to from the study? Could you comment on that? Or when the patients actually fasted, how long did they fast for?
It was 12 hours from the night before.
Okay, so essentially it's like they're having breakfast. Would you recommend that if persons are supposed to consume a certain type of food when they wake up, it's better than having the carbs? Would it have the same effect in terms of producing the amount of gas? I don't know, that's my question.
I have to begin by saying I'm not qualified to answer that, unfortunately. My studies have not extended very far into the breakdown of fats after fasting. But what I would say is that I've had a similar question from an audience of gastroenterologists, and he asked me, should I put dried fruit on my cereal or should I put sugar? Because obviously dried fruit will produce a lot more fructose that would be unabsorbed and therefore create that watery condition, whereas putting sugar on your cereal wouldn't create that. So with my unqualified response, I would say that the sugar to stay away from after a night fast would be fructose, but I can't comment about whether having carbs or fat would be preferred, unfortunately. I'm sorry.
Okay, thank you for that.
Okay, HGL, you may go ahead.
Good morning. I'm Heather Lamb. I love the presentation. It was a lot of the science is over my head. I'm not a scientist at all. But I really enjoyed the presentation and I found it very interesting. My question has to do, I noticed you mentioned healthy adults and you just said healthy adults, but I wondered if you considered using postmenopausal women as a delimitation factor, or if you did anything that had to do with older adults, because you didn't mention the age of the healthy adult participants. So I'm wondering about that.
So that's a really good question. And I think in some respects, because we're operating out of a university, that our cohort is biased towards a younger set of participants. So most of our participants would have been between the ages of 18 and 25. Some of the oldest ones would have been probably 30, which is obviously quite young still. What we noticed was that in our patient studies, the cohort was normally over the age of 50. So that would have been the more of the menopausal region that you speak about. But I have not actually done a study where we recruited healthy postmenopausal women. And that would be quite interesting. Unfortunately, we do find that it is that cohort which tends to suffer more from irritable bowel or functional bowel disorders, but it would be very interesting to see a healthy cohort of older participants, but we've not done that, unfortunately.
Okay, ladies and gentlemen, thank you, Heather. Heather, that's HGL. I want to acknowledge Catherine, another of Dr. Pinnock's daughters, who I saw popped up somehow. I don't know if she's still here. I don't know if Heather and Georgina are all here, but I'm sure the news will reach them, Catherine, of how well you are doing and how much their father has influenced who you have become and what you are now able to do for the world. Dr. Sadler McKnight said she had to leave, but your work is quite significant and she's looking forward to the collaboration with you and Dr. Pinnock Jackson. And she also knows several children with these issues. And Catherine, you should keep up the good work. Ladies and gentlemen, I think it is time for us to bring this wonderful show certainly to a pause. It will continue because others of Dr. Pinnock's students will speak in seminars that are to come. At this point, I am going to invite Dr. Wilson to give the closing remarks. Dr. Wilson.
Thank you, Dr. Wilmot. I'm almost moved to tears for many reasons. That we could have all come together like this to honor Dr. Pinnock. He has, my apologies. Sorry, you know I don't usually get speechless.
Oh, Ken, you're making me cry. Just move it on, move it on.
But we certainly saw the Pinnock method shining through in your presentation, Catherine. The detail, your attention to detail, how you spoke, how you explain things, and I think Dr. Pinnock would be extremely, extremely proud of you this evening, this morning, as we all are. Keep up the good work. You are making chemistry, UWI Muna, extremely proud. A lot of our lecturers are here today, Dr. Reese, Dr. McKnight, Dr. Cates, Dr. Roy. I will not call everybody, our colleagues, Nadine, so here, Anchor. You have gone abroad and you have represented well. And I'd just like to say congratulations, keep up the good work. And Dr. Pinnock would have been proud, and we are indeed standing on the shoulders of a very, very great man. Thank you for this opportunity, Dr. Wilmot.
Thank you very much, and yeah, thanks.
Thank you, ladies. Catherine, if we were in C2, I would have a gift for you. I would give you a hug. I hope you can feel the love, and certainly when we see you, the department will have that token ready to hand to you. Colleagues, friends, stay safe. We continue to shine. See you next week.
Bye. Thank you.